Now that we've talked about the nucleus, and the last comment we'll make about it is the following: virtually every eukaryotic cell has one, except for red blood cells, but recognize that there are certain cells that have more than one nucleus. Hepatocytes would be one of them. They're frequently binucleate. Osteoclasts, which we'll see when we get into bone marrow, they're typically multinucleate, as are megakaryocytes. All right. The other membrane-bound organelles that we're going to spend time on, functionally as well as anatomically, are the elements of the endoplasmic reticulum. It comes in two flavors, rough and smooth. One of those, the rough ER, is closely associated functionally with the Golgi apparatus. And the Golgi apparatus has some functional relationship to lysosomes, which we'll see. But we'll distinguish lysosomes from peroxisomes or microbodies, and finally all of those from the other double membrane-bound organelle, which are the mitochondria. All right, so first, before we do that, let's look at an overall schematic view of a typical eukaryotic cell, and it shows quite nicely the configuration of the nucleus. You can see forms of chromatin schematically illustrated as being coiled up, heterochromatin, since you're visualizing it. You can see that the nuclear envelope is a double membrane configuration with pores, and you note that the nuclear pores are directly continuous with elements of the smooth and rough endoplasmic reticulum. Not labeled, although certainly prominent, is a nuclear inclusion, which is certainly the nucleolus, the little nucleus that we identified earlier. Now, we're going to proceed by talking about the elements of smooth versus rough endoplasmic reticulum. You can see in this schematic configuration that they consist of parallel arrays of membrane-associated cisternae. The difference in them anatomically is the rough ER is characterized by the presence of electron-dense dots attached to its membrane components that make it rough, and the dots basically, as we'll see, are bound polysomes. The smooth endoplasmic reticulum looks very much like rough endoplasmic reticulum, but without the electron-dense dots. Next, note that smooth endoplasmic reticulum, particularly in this picture, looks very similar to the Golgi apparatus. Very different functionally, but we're going to have to distinguish them very carefully in different electron micrographs, and we'll do so. Then, certainly the smaller of the membrane-bound organelles, also illustrated in this picture, are mitochondria, lysosomes, and peroxisomes. And then finally, this also gives us an indication of what some of the cytoskeletal elements are. We have three flavors of those. Cytoskeletal elements come in the form of microtubules and two forms of filaments. One class are known as intermediate filaments. The other class are known as microfilaments. And then, although not labeled on here, a couple of the other features of cell biology we need to discuss are how cells are related to additional cells by junctional specializations, and finally, how cells have certain luminal surface specializations of a wide variety of types that underlies a different functional specificity. Next conversation deals with ribosomes. Ribosomes are composed of ribosomal RNA and protein, and they have a combination of large and small subunits. They are assembled in the nucleus and transported to the cytoplasm through the nuclear pores. Frequently, the ribosomes will form polysomes, which consists of a single messenger RNA that's being translated by several ribosomes at the same time. The important point about these polysomes and ribosomes, they exist in two forms. The bound polysomes are those that are attached to and functionally associated with the endoplasmic reticulum. The free polysomes are situated in isolation in the cytoplasm and not associated at all with the endoplasmic reticulum. But here's the point. Recognize that free and membrane-associated or bound polysomes each produce three different kinds of proteins. Arguably, the more important array of proteins are produced by the membrane-associated or bound polysomes that are associated with the endoplasmic reticulum. And important because those three types of proteins are secretory proteins, membrane-associated proteins, and lysosomal hydrolase enzyme proteins. The proteins produced by free polysomes all stay within the cell, unlike some of those produced by the membrane-associated ones. And there are 3 different proteins produced by these unbound polysomes. They are those that are to be incorporated into the nucleus, incorporated into peroxisomes, or are associated with mitochondria. Next, what does rough endoplasmic reticulum look like in the electron microscope? As shown well in this particular micrograph, it is a series of parallel arrays of cisternae that have the same diameter end to end. In other words, the form of them is the same end to end. They are parallel, and again, they're easy to recognize because they are characterized by the presence of electron-dense dots, which represent membrane-associated polysomes. Smooth ER, on the other hand, looks like rough ER, but lacks the bound polysomes or ribosomal association. And one of the functions of basically smooth endoplasmic reticulum, its major thing is to produce steroid-based hormones, but it's also associated with a wide variety of other biosynthetic reactions. So first off, let's compare the two. Note that in this electron micrograph, note how regular and uniform the structure of rough endoplasmic reticulum is. Not only are the cisternae basically the same diameter end to end in an individual cistern, but note that the cisternae are almost equally spaced from one another in this electron micrograph. In contrast, smooth endoplasmic reticulum has a much more irregular array of its cisternae, nowhere near matching the regular pattern that's seen in the adjacent rough endoplasmic reticulum. And using the pointer, we can see other membrane-associated organelles. This happens to be from corpus luteum. So the mitochondria have a kind of a Christie-like tubular matrix in them. So these are all membrane-bound mitochondria down here. The electron-dense structures could be microbodies, could be primary or secondary lysosomes. It's kind of hard to tell based on their configuration without specialized staining. Now, what else is smooth ER doing? We identified its involvement in steroid hormone synthesis. It's also involved in hydroxylation reactions, detoxification reactions, mainly by using a whole array of enzymes that functionally are associated with cytochrome P450. And in addition, smooth endoplasmic reticulum is also involved in conjugation reactions. Lastly, it's involved in glycogen metabolism, involving degradation in gluconeogenesis, lipid metabolism, and as we'll see in striated muscle cells, skeletal and cardiac muscle, it serves as a sequestration site for calcium ions that are closely associated with the cell membrane of skeletal and cardiac muscle cells. Next, in this electron micrograph, we have a view of the Golgi apparatus. And note that Golgi apparatus looks very similar to smooth endoplasmic reticulum in that it consists of parallel arrays of cisternae. The difference in the electron micrograph is that the Golgi apparatus typically has a slight curvature to it. So it's going to have a convex face and a concave face. And second, at the ends of the cisternae, the parallel cisternae, frequently we see, and we certainly do here, dilatations or blebbing on the ends. So unlike the smooth endoplasmic reticulum, if it were an irregular pattern, the Golgi apparatus cisterns do not have a uniform diameter from end to end. Now, what does the Golgi apparatus do? Well, it is closely associated with the rough endoplasmic reticulum, because each of the 3 kinds of proteins that are produced by the rough endoplasmic reticulum must next be sent to the Golgi apparatus. And you can see the proximity of the cis face of the Golgi apparatus. That's the entry point of all 3 of the proteins newly synthesized by the membrane-associated polysomes of the rough endoplasmic reticulum. So the medial Golgi is a site where those proteins begin the sorting and glycosylation process. And by the time the proteins reach the trans face, the packaging of them has occurred after sorting. And if they're going to be packaged for secretion, typically the newly synthesized protein will be incorporated into a membrane-bound secretory vesicle that will then move and fuse with the extracellular membrane of the cell and the secretory granules be extruded by exocytosis. Now, two clinical scenarios basically involve the Golgi apparatus. One is hyperproinsulinemia. This is characterized by elevated levels of proinsulin in the serum because of an enzyme defect, a peptidase defect that fails to cleave proinsulin to insulin and a related C-related peptide in the Golgi apparatus. And the clinical manifestations of this are similar to patients seen that have non-insulin-dependent diabetes mellitus. The other Golgi-related disease is known as inclusion cell disease or eye cell disease, named for dense inclusions to typically are present in fibroblasts. This eye cell disease is again caused by an enzyme defect. Specifically, it's a phosphotransferase enzyme defect that fails to adequately mark hydrolase enzymes for incorporations into lysosomes. Because as part of the sorting and marking process, those hydrolase enzymes have to be phosphorylated. And the phosphate basically is added in a two-step process that involves that phosphotransferase enzyme. That enzyme typically is deficient, so what in essence happens is there's tremendous inclusion bodies that occur in fibroblasts, and basically the lysosomes lack their appropriate hydrolase enzymes. Clinical features of this involve multiple skeletal abnormalities and a lifespan of typically less than 10 years. Next, speaking of lysosomes, lysosomes are our next membrane-bound organelle, and as such they contain very low pH hydrolase-based enzymes that are required for intracellular digestion of large macromolecules. Lysosomes generally come in two forms, although the distinction between them is not as precise as it used to be. Classically, a primary lysosome has not acquired materials to be digested and has just been formed by budding from the transphase of the Golgi apparatus containing its hydrolase enzymes. A secondary lysosome is basically involved in some aspect of the degradation process and typically has some sort of interiorly located, partially degraded substrate. Lysosomes comprise or contain about 60 hydrolytic enzymes. All virtually are acid hydrolases, but again, these hydrolases are produced in the rough endoplasmic reticulum. They have to be transferred to the Golgi where they are modified by phosphorylation, sorted, and packaged ultimately into lysosomes. As you'll hear in pathology, there are a number of lysosomal-based storage diseases based on various hydrolase enzyme deficiencies that result in deficient breakdown of sphingolipids in the clinical conditions known as Gaucher's, Niemann-Pick, and Tay-Sachs disease. Finally, an electron micrograph view of primary and secondary lysosomes. Probably the homogeneous electron-dense structures toward the middle of the picture are most likely primary lysosomes, and the structures at the tip of the arrows are more likely secondary lysosomes that contain partially digested substrates. Now, how do lysosomes differ from peroxisomes? Peroxisomes are really a specialized kind of a lysosome. In a way, they're frequently referred to as being microbodies, and like a lysosome, they contain a number of enzymes that are involved in various stages of degradation. But in this case, the major toxic product that's being degraded by peroxisomes is hydrogen peroxide. And the major enzyme, therefore, that's a component of peroxisomes is catalase. Specifically, its job is to degrade the hydrogen peroxide to water and oxygen. But peroxisomes are also involved in the oxidation of long-chain fatty acids, phospholipid exchange, bile acid, as well as cholesterol synthesis. And peroxisomes are unique in that their enzymes, including catalase, as we said, are proteins that are basically produced by unbound or free polysomes within the cytoplasm. These enzymes that end up in a peroxisome do not have to pass through the Golgi apparatus. They're simply incorporated directly into the peroxisomes. But peroxisomes may lack one or more of their catalase-based and other enzymes, giving rise to a number of peroxisome biosynthesis disorders, or PBDs, the most severe of which gives rise to what are called empty peroxisomes on exam questions. And this is Zellweger syndrome. And the mildest form of a peroxisome biosynthesis disorder is Refsum disease. And again, you'll distinguish some of the signs and symptoms of these in pathology. Finally, we see a view of a peroxisome. It's kind of difficult without specialized staining techniques to visualize. It's kind of shown here on the right side of this electron micrograph. But it makes a good comparison between a membrane-bound kind of amorphous and relatively homogeneous structure, such as a peroxisome or a primary lysosome, compared to that of our last membrane-bound organelle, and these are mitochondria. Mitochondria, like the nucleus, are the only other subcellular organelles that contain two membranes. And certainly mitochondria are very distinct in that their inner membrane is thrown into folds known as cristae. And certainly the major job of mitochondria is to synthesize adenosine triphosphate, or ATP. They are capable of making their own proteins, and they have their own DNA as well. And it's important to recognize where the ATP synthesis and the oxidative phosphorylation are taking place. First, on this picture we've shown before, we can visualize quite nicely the Golgi apparatus, but although we didn't allude to it before, you can see multiple examples of the hot dog-like structures known as mitochondria, and you can see how evident and obvious the internal cristae matrix of the interior is. Note again that the real business end of a mitochondria is its inner membrane. It's thrown into numerous foldings or infoldings called cristae that dramatically increase the surface area of a mitochondria, and they are the actual sites that contain the enzymes associated with electron transport and oxidative phosphorylation. And one clue as to the metabolic activity of an epithelium or a tissue is based on the presence of not only the number of mitochondria, but the number of cristae per mitochondrion, which is largely proportional to the metabolic activity of the cells in which they reside.